Hello, my name is Keretz, and today I'll be talking about species distribution modeling for native and invasive mussels in the St. Clair and Detroit rivers. Native freshwater mussels, or unionids, are a macroinvertebrate bivalve that contribute multiple ecosystem services to our environment, such as water filtration, nutrient cycling, and sediment stabilization. North America hosts the highest diversity of unionids worldwide, with approximately 300 species across the continent. And in the Great Lakes region, there are approximately 43 species in Michigan and Ontario. The St. Clair and Detroit rivers are in the Huron-Erie corridor of the Great Lakes. And these rivers form the international border between Michigan and Ontario. Additionally, these rivers are part of the Great Lakes shipping channel, and both of these rivers historically supported approximately 39 unionid species, including the now federally endangered northern riffle shell and snuffbox mussels. However, unionids were considered extirpated from these rivers shortly following the introduction of zebra and quagga mussels, collectively dreissenids, into Lake St. Clair in the 1980s and the 1990s, respectively. Dreissenids are particularly harmful to unionids because they could congregate on the unionid at high densities. This is known as dreissenid fouling, which can interfere with the unionid's ability to feed, respire, move, and reproduce. In the Great Lakes region, dreissenid fouling has been a major cause of unionid mortality. This, in addition to other harmful factors, such as habitat degradation, are why unionids today are considered one of the most imperiled faunal groups in North America. Fortunately, there is reason to hope. Unionid refuges, or places with living unionids and little dreissenid impact, have been found in the Lake St. Clair Delta and in Lake Erie coastal wetlands such as Metzger Marsh and Crane Creek Marsh. Due to their highly imperiled nature, it has become increasingly important to identify and protect the unionid populations in these spatial refuges. One method for locating unionid refuge space is modeling known refuge locations. Boston-Browick et al. Were able to successfully predict unionid presence in Lake Ontario using Maxent, unionid presence locations from Lake Erie, and variables like fetch, shoreline geomorphology, and depth. Following up to this study, we are asking whether unionid presence can be similarly modeled in large river systems, particularly the Detroit and the St. Clair rivers. This leads me to my study objectives. The first component of this study was to determine the relative abundance of any remnant unionid populations in the Detroit River. We then used the unionid presence data from the Detroit River to create predictive species distribution models for the St. Clair River. In the third component of this study, we surveyed the St. Clair River in order to validate the predicted species distribution models and to determine the relative abundance of any unionids remaining in the St. Clair. So, starting with the Detroit River, our sampling took place in summer of 2019. We started with a selection of 60 sites that were a collection of randomly selected, historically surveyed, and potential refuge sites. Once we arrived at a site, we surveyed for unionid mussels by a 1 person-hour time search using scuba. We also surveyed for dreissenid mussels by collecting 6 petite ponar grabs at each site. During our summer of sampling, we surveyed a total of 56 sites throughout the Detroit River. At 39 of those sites, represented here by the white circles and white triangles, we collected over 2,000 unionid shells representing 31 species. Most excitingly, we did find live unionids at 5 sites in the lower half of the river, represented here by the yellow circles. Among these 5 sites, we found a total of 220 live unionids of 11 species. Now, our Detroit River work is not the focus of today's talk, but if you're interested and would like to know more, please check out our newly published manuscript detailing this work. So now that we've got data from the Detroit River, we used those 5 unionid presence locations to create species distribution models using Maxent. The environmental variables we considered were depth, total velocity, east-west velocity, north-south velocity, river distance to the river's inlet, outlet, and the nearest tributary. Finally, we used a potential distribution model to estimate dreissenid presence throughout the rivers. Within Maxent, I split the presence data into 80% training data and 20% test data. Each model creation went through 10 replicates, using the subsampling and random seed parameters to ensure that a different training dataset was used for each replicate. The models were finally created using the default cloglog output. The predictive species distribution model for unionids used 4 environmental variables: total velocity, river distance to the outlet and nearest tributary, and estimated drusenid presence. These provided the best model with an AUC of 0.959. We then projected the model onto the St. Clair River. This model assisted with site selection, and these pink boxes, or model selected sites, were selected in areas that the model thought unionid presence would be likely. In addition to these model selected sites, and similar to how the Detroit River was surveyed, we added a collection of randomly selected, historically surveyed, and potential refuge sites for a total of 79 potential sites to visit. We set out to sample the St. Clair River last summer on July 19th. Like the Detroit River, we used a 1-person-hour time search by scuba to survey for unionids, and we collected 6 petite ponar grabs at each site to estimate drusenid densities. We were able to visit 51 sites throughout the St. Clair River. Among 33 of those sites, represented on this map by the white circles, we found 479 shells representing 23 species. Additionally, we did find live unionids at 7 sites throughout the river, shown here by the yellow circles. Among these 7 sites, we found 14 live unionids representing 9 species. And as a brief side note from an additional part of this project, here is an estimated distribution of live drusenids throughout the St. Clair River, with the smallest circles representing sites with 0 live drusenids per meter squared, and the largest circles representing sites with over 5,000 live drusenids per meter squared. The light blue represents live zebra mussels, and the dark blue represents live quagga mussels. As you can probably see, a majority of the drusenids in the St. Clair River are indeed quagga mussels, since only a few sites had any live zebra mussels. So by now you're probably asking, well, how did the model do? Well, live unionids were found at 4 randomly selected sites, 1 historically surveyed site, 2 potential refuge sites, and 0 model selected sites. So, in other words, the predictive model created from the data collected in the Detroit River failed to predict unionid presence in the St. Clair River. Now for the big question. Why did the model fail to predict unionid presence? To explain my hypothesis, please remember the 4 variables that were used to create the Detroit River model: total velocity, river distance to outlet and nearest tributary, and estimated drusenid presence. Just looking at the total velocity variable, the mean velocity where unionids were found in the Detroit River was 0.3 meters per second, while the mean velocity where unionids were found in the St. Clair River was 0.5 meters per second, meaning we found live unionids in the St. Clair River at faster flows than where we found them in the Detroit. Additionally, looking at that second variable, river distance to outlet, if you'll recall, all of our live unionids were found in the lower half of the Detroit River. And as you can see in the St. Clair, we found live unionids all the way at the top and all the way down at the bottom. So where do we go from here? Well, our first step is to use the 7 unionid presence sites from the St. Clair River to create a predictive species distribution model for the Detroit and assess that model using the data we collected in 2019. To do this, I used the same environmental variables as were used before, and as a brief reminder, those were depth, total velocity, east-west velocity, north-south velocity, river distance to the river's inlet, outlet, and nearest tributary, and finally, estimated drusenid presence. The predictive species distribution model for unionids in the St. Clair used 5 variables: estimated drusenid presence, river distance to the river's inlet and nearest tributary, and east-west and north-south velocity. This gives us the following model with an AUC of 0.949. When we project the model to the Detroit River, the model predicts unionid presence up near Belle Isle, which is quite the opposite of what we really found. Though I will say this model has been improved upon since our first attempt, which is something to note and supports further potential improvement. What we've learned is that the models made so far are highly predictive for their respective rivers, but do not predict well in other systems yet. So, here are some ideas for improving the model for future use. First, I have some new environmental variables I would like to add, such as distance from hardened shoreline or to the nearest dock. I will also be creating models using unionted presence locations from both the Detroit and St. Clair rivers to create a model for the whole system as opposed to just one river at a time. There's the potential to explore other model types besides Maxent, such as GARP. And finally, we do plan to continue our surveys in both rivers this upcoming summer in order to visit some areas highlighted by the Detroit River model and to hopefully visit some areas that were missed during our initial round of sampling. All of this work contributes towards our overall goal of understanding the distributions of unionids in the Detroit and the St. Clair River systems, which could contribute towards unionid conservation and their management in large river systems in the future. So with that, I'd like to thank our funding source and all of our collaborating agencies. It does take a small army of people to accomplish this work, so I'd like to thank every single person on this list who has been helpful and supportive, and I thank them for their continuous dedication to this project's success. If you have any questions or comments, you can reach me during the scheduled question period or at the email below. Additionally, if you would like to keep up with this work, you can follow myself and my advisors on Twitter.